Today I'm going to talk about crowdsourcing research materials, and I'm sure you can imagine that the quality of those materials is what's really of paramount importance to the scientific community. I'm going to talk about how that's influenced our COVID-19 distribution, which is ramping up really quickly, not a surprise there. And I'm also going to just tell you a little bit about Addgene in case you're not familiar with what we do. So Addgene is a nonprofit, and our mission is to accelerate research and discovery. And we do that by helping scientists access research materials, so physical materials. We have a physical collection here in Watertown, Massachusetts. And of course, all of the important data associated with those materials, because what good is a plasmid if you don't have a really good quality sequence and information about its cloning? And so we collect materials from the research community, both primarily academic institutions, but also, as I'll mention, industry deposits to Addgene, and then we distribute those out to scientists around the world. And we actually have quite a large collection, and I'm going to talk about just these topics here to introduce you to the collection at Addgene and the work that we're doing now. So Addgene has mostly plasmids. We have over 80,000 of them now, individual. So before you make something, it's probably quicker to get it from Addgene. Most places in the world can get requests from us in a couple days. We also have kits of plasmids, which of course bring the per plasmid costs much cheaper. Pooled libraries that are incredibly useful for screening. Someone else may have already created the library that you need for your experiment. Viral vectors, both AAV and lentiviral prepared vectors ready for experiments. See our website for the huge catalog and useful catalog there. A lot of that AAV work is in neuroscience, and a lot of the lentiviral work is, we have some COVID-19 materials, and we also have some CRISPR genome engineering related libraries. And also pooled libraries, viral vectors, already ready for you to transform and do an experiment. Very exciting. So, a lot of what we do is we have an incredible distribution system. And so we get, right before the crisis, the COVID-19 pandemic, we were distributing greater than 750 items each workday to scientists all over the world. We just hit our 100th country. Tunisia was country number 100. And we do that with a lot of export-import support, you know, something that is very hard for you to do from your lab. We can ship anything, anywhere, pretty much. And so that is part of the value that we bring to the collection. It's great to contribute to the collection because, you know, that's probably your freezer on top and that's our freezer on the bottom. Everything's barcoded. We don't lose anything. You'd be surprised to hear how many scientists request their own materials back. Also, materials distribute often more than people expect. You may think that no one is interested in what you're doing, but other people want to use your materials and, of course, cite you. And the scientists actually experience an increase in citations when they deposit to the repository because people use their materials and then cite their work. If you're at a company, you might want your technology to get exposure in the academic community especially. That's another great reason to share materials. So we're able to distribute worldwide, as I mentioned. Here are the places that Addgene has distributed. We can get materials, you know, if you give them to us, we can get them anywhere. So it saves you a lot of headache, especially for things that are popular or requested a lot. We use distributors in some countries. That helps things both with language support and payment support and also distribution support. And that has been a real advantage. We also have a very small office in the UK. Shout out to our international Addgenes. And so if you're in the UK and you want a little more exposure to Addgene, they can arrange a visit. So we also, one of I'm leading up to the fact why SeqWell has been so important in our process. Of course, Addgene doesn't just share the materials. We share an enormous amount of information related to these materials. So our website is actually an information and education portal. We get over 2.5 million page views per month. I'm sure you can, anyone who knows anything about the internet knows that's a really big amount for just a single website. That's not Amazon. So people call us the Amazon of plasmids, and we like that. So, you know, we have many, many unique users each month, and people are accessing not just to order, but also for all of the educational information and support and protocols and data that we are able to provide. And the Addgene, being a nonprofit, is really our mission to make this stuff useful if it makes science faster for you. And that is how we focus our work. So every product page has an enormous amount of information and links and useful specifications. And everyone shares. So you can, a person who deposits can share protocol tips or information or caveats about their material through the product page. And then there's, of course, all kinds of other information on the website. We have a very popular blog. And if you're interested in writing for the blog, we would love to hear from you. And the blog has protocols, information, unexpected tips, summaries, all kinds. If you're trying to stay up with molecular biology, it's great to follow the blog. We also have some career stuff, which is really useful and incredibly well written by many, many great scientists. We have these protocol pages that are viewed a lot. We actually have a lot of protocols on YouTube in video, especially very basic for very entry-level people in the lab. So how to pour plates, and also some more senior, some more advanced protocols, if you will, like how to do a virus prep and get a good band of virus purification. So that's a great place to look if you're doing training for your students. We have a fantastic technical support team. These scientists answer the phone, and then our customer support team helps you make sure that your package is where it's supposed to be. And they answer thousands of questions every year, and love to do that. That's part of our mission to help you. We also have these educational resources. We don't yet have a COVID-19 one, but we do have a COVID-19 website page that can help you find materials quickly and information. So now to the quality control, which is so absolutely vital to a central repository. The identifying the materials as best we can is important in adding the value. It's what makes people want to request from us. Why not get validated material when you are accessing materials that are being shared? So sequence really matters for plasmids, and plasmids is what we share the most, although of course plasmids are the basis for all of our products, the kits, the libraries, the viral vectors. So it's really, really important that anything there is sequenced. And you know, I'm sure you know, you have horror stories. I have my own from when I was in the lab for some 25 years of that plasmid that you tried so hard. And you know, back in the day when we didn't sequence everything, when it was all expensive and hard and we had to sequence on a radioactive gel, you know, we didn't have that sequence, and you could go down a really scary rabbit hole of failed clonings because a site was missing or the sequence was incorrect or you had an unusual variant. It's just like a scientist's nightmare not to know what they're working with. So it is our job and our pleasure actually to really provide people with good quality materials so that they have fewer problems with their experiments down the line. And so this is our quality control team, and they're all, of course, working. Most of them are working from home, occasionally coming in, you know, we are intake of plasmids, in fact, is accelerating in this crazy time. We're getting some great deposits, and they're constantly monitoring. And then I'm going to tell you about the huge COVID-19 collection that they are quality controlling and making sure it's going well. So we are so grateful to them for their hard work, even at a really tough time. So here was the challenge. We went to multiple vendors with a plate of our plasmids because we were doing Sanger sequencing, which meant we could only afford to get, because we're a nonprofit and we keep prices as low as possible, we could really only afford to get a small piece of validating sequence from each plasmid. And that meant there could be mistakes in the backbone that we didn't know about, or other places in a large plasmid. And we did our best to sequence across mutations to make sure that they were present, and wild type and mutation variants. But it really was, in order to afford the model that we are doing, we really needed a cheaper way. When I first came to Addgene 10 years ago, high throughput sequencing and next-gen sequencing was becoming more usable and useful, but it was interesting. It didn't work well on plasmids. So all of the algorithms and many of the processes were designed for long, you know, genomic sequence, but didn't really work on small circles of DNA with a lot of repetitive elements. And so we have this challenge plate, and the challenge plate was, we admit, an extremely difficult to sequence plasmid. A lot of repetitive elements, viral elements, things that were large, things that were too small, and things that we didn't have reference sequence for. So de novo sequencing of plasmids, which is sometimes the case. People deposit and they don't provide us with a reference sequence. So with only one exception, all the companies that took on this challenge were not able to successfully provide us with circular sequence at the end for the majority of the plasmids on the plate. And given the diversity of what's coming into the repository, that's what we needed to make this a go. So we had a meeting with SeqWell. We were introduced by mutual friends and we were excited. They seemed like, yep, we can do this. We gave them the plate. And to be honest with you, they did not succeed. So it was only about a 60% assembly, and some of those were, even some of the easier ones didn't get assembled. It's not good enough with the throughput that we're doing. We're doing plates a week at this point. So, but Joe was tenacious and said, darn it, this must work. We are going to figure this out. And that is in fact what they did. So they tested and optimized both the algorithm and the procedures until we had a very high rate of successful sequence and assembly to circular plasmid. So much so that it was going to save our scientists an enormous amount of time and provide much higher quality sequence at a price that Addgene could afford as a nonprofit, that made the value of these plasmids increase so much that it was worth this investment for us. So big success story as far as networking, because I'm so delighted that this is working out. It's been, it's helped tens of thousands of scientists, hundreds of thousands probably, with better quality materials. So what we were doing before, as I mentioned, was Sanger sequencing, covering a small portion of the plasmid. After that, we could reduce the time to full sequence for less than 5 weeks. And this doesn't seem like a lot, but 1.25 minutes saved of time per plasmid in analysis is more than 5 hours a week for a single QC scientist. And again, as a nonprofit, for any organization, saving that time and that efficiency for even better data was a huge win for us. High throughput, reasonable speed, best quality validation of plasmids has been enabled. And that's where we entered into this COVID-19 crisis, supporting that crisis, being able to do that so quickly. I'm sure you all know how quickly the scientific community mobilized. And within a couple weeks of the initial shutdown in Massachusetts, we were back open, starting to take in materials related to COVID-19 research. So that was a little scary, because first we had to close and figure out how we were going to work in this new normal as required personnel. I'm sure any of you that are working in the lab are going through the same thing right now. It's quite challenging. But you know, it was our duty. We feel it was our duty to really support the research community to do this research as quickly as possible. So the Addgenes were back in the lab. Here's a couple of our lab technicians who are involved in actually picking from the collection. That's behind Shannon in the back there. That's the Addgene collection. I'm here on site alone in my office. They're off in the lab. I'm a couple hundred feet away from me. We don't see each other much, but we're getting an enormous amount of work done. So we mobilized quickly. The plasmid collection is essentially being updated daily. You can look on our website for easy links to see what's coming in and what's available. I'm going to talk a lot about the fact that things are available quickly and available for industry requests. We're also providing protocols and resource links. And we're also collecting information on what's needed but not yet available. So you can get in touch with us for any of those reasons. And Addgene is happy to work with you to see if we can help you find what you need or help you deposit what might help others in accelerating their research on coronavirus. So very quickly, was our first day. We were opened up for shipping again, was March 23rd here in Massachusetts. And this is a little bit outdated already. We've actually distributed over 5,000 COVID-19 related plasmids to date. And on the Addgene website, when material has been distributed 100 times, it gets a little blue flame. The depositor gets an award for that. They love those things. Actually, it's here. I can show you one. I had one on a slide. This is what the Blue Flame Award looks like. They're quite coveted. You can only imagine how many citations and how much prestige that is for the depositors in really supporting the full scientific community. And so a lot of these plasmids, you know, pretty much in a couple weeks had blue flames, were incredibly popular. Many, many people are studying coronavirus mechanisms now. So we really needed to get depositing working fast, even faster than usual. So we combined some rapid Sanger sequencing with the SeqWell CPS approach to make sure that while we were speeding things up, we still maintain that excellent quality. And so that allowed us to get things online quickly and full sequence in 18 days, which is really quite fast for a whole quality control intake procedure. We have to make our stocks, we have to validate them, and we're doing thousands of these at one time. Also, things that didn't pass the Sanger, already the SeqWell sequencing was in process, so we didn't lose any time, and that allowed us to really move things faster. We also, I hope, if you're working on coronavirus, I hope you know about the Ginkgo Bioworks deposit. They're in the process of depositing more than 3,000 tagged COVID-19 related plasmids, tags, proteins, different kinds of useful things for different organisms expression, yeast, E. coli, and mammalian. And this was all made possible with the SeqWell complete plasmid sequencing to add confidence to these things that were rapidly synthesized by Ginkgo. So COVID-19 is awful, it's horrible, but there has been one teeny tiny silver lining that we have seen here at Addgene. And that is that plasmids that are being deposited for this research have been available, many of them, I think almost over 90% now, are available for for-profit requesters. So in the past, Addgene mostly only distributed the majority of the collection to nonprofit universities and nonprofit organizations. And we've been wanting to bridge that gap for a long time. And I think universities showed an unprecedented amount of common sense in saying this isn't a time to take a long time with the legal work and prevent distribution. So apparently science is made more open by a universal health crisis. That's good to know, good common sense on the part of the world. So since March, I would say it's more than 1,000 now. It's thousands of new plasmids related to COVID-19 are available for request. And 90% of them are available to for-profit industry-based scientists anywhere at any company. And you can look at our industry page for links to the materials that are available to industry. That we still have a lot of things that aren't available. We're working on it. But if you start there, you'll really be able to find out what is available to you. Of note is both the COVID and SARS-CoV-2 collection. The Resolute Consortium and Structural Genomics Consortium, Michael J. Fox Foundation has always deposited fully open. But also, this is the Michael Davidson Fluorescent Protein Plasmid Collection, among other useful plasmids from Florida State that are all available to industry. And it's quite a useful collection. So if you use plasmids in your research, you might want to take a look. So kudos to these depositors. These are the academic institutions that to date have made their materials available both for-profit and nonprofit use. And we're really excited about bridging this gap. In addition, companies are also depositing to Addgene. And these are some of the early shares. These are for-profit organizations that shared materials early. So not just in this last crisis, but in the past have shared through Addgene. Materials that are quite useful to scientists and quite popular. And they do that for a variety of reasons. Some of them want to get their exposure for their company research in the basic research community. Their future employees are coming from those organizations, from those universities. And also, they may have plasmids in the freezer that are no longer used or useful. It's a shame, they're a waste. It's so much faster for science when these things are shared. They may want to show the value of the technology. So if you're to buy tech and you want other people to test your technology because it would be useful for therapeutics and you want more data on that, you might want to share your materials through Addgene. Also, if you are distributing things anyway, and it's not your company's practice to share small boxes of low-cost plasmids all over the world every day, it may be easier to have them shared through us because that is our business. And of course, you have records of every distribution if you own the materials. Every depositor at Addgene has their own web page with all of their own records kept there, easily accessible so they can see who's requesting, because we like to start collaborations. And sometimes that happens, so it's great. So Addgene's mantra is, how can we help? We're here to help you. We have many social media streams, but in particular, the blog and our YouTube sites have incredibly useful material. And we are happy to take your questions at this time. So let me know if you have any questions, or please reach out directly to us at help@addgene.org. Okay. Okay, so I don't see a lot of questions. So I am going to turn it over to Joe. Okay.
Thanks, Joanne. I really appreciate hearing about the work that Addgene is doing, and we certainly are excited to continue to help with the plasmid QC and validation pipeline that we built with you. It's certainly great to see those tools that the community is building still being circulated and distributed to the world during the COVID-19 shutdown.
Absolutely, and even more so.
So I'm going to go ahead and share my slides here and tell you about the work we're doing here at SeqWell. I really appreciate everybody who's joined us here today. I think certainly building on what Joanne said, I think we see opportunities to really provide tools that will allow us to support COVID-19 research in various ways. And in this case, I think, certainly in looking at the application of high throughput genomics for sequencing of the SARS-CoV-2 virus. So to talk through the outline here today, the talk is really going to focus on sequencing approaches to SARS-CoV-2, and I'm going to give you a little bit of background into our approach on that. Part of that will be introducing the PlexWell technology that we have built our approach on, and how we've integrated that with the Arctic network amplicon protocol that was created to support multiplexed PCR and sequencing of different type of viral samples. From there, we'll go through a SARS-CoV-2 sequencing case study that we built internally here, and we'll talk about some of the performance features of that we think will be interesting for labs around the world who are interested in sequencing coronavirus samples. So at SeqWell, we really focus on making library prep tools that can help accelerate genomic applications and across a range of different problems. We think of library prep really as effectively the software of the genomics era, a critical tool for really matching the capabilities of what sequencers can do to the research and clinical problems that people and researchers are looking to solve. Many of those applications, as I'm sure many of you are aware, really have a lot of common challenges that they are encountered. For example, workflow, scalability, and getting better data out of the back end. And really, we think of library prep as being the key tool for solving some of those challenges, and thinking about ways of creating library prep tools that will improve the performance of sequencing by creating, for example, more integrated, better workflows that are simpler to execute, can process more samples, and give better information out at the end. As you heard a little bit from Joanne, our history and partnership with Addgene is certainly part of how we have built some of those tools. Going back a few years, we started working with Addgene really in how to, as Joanne mentioned, create a robust and scalable NGS-based plasmid QC and validation pipeline. Certainly part of that was based on, what is really a critical issue is that in order to do that with NGS and convert from what was previously a Sanger-based pipeline, we really had to address kind of some of the real key issues there. For example, large variations in plasmid content, and the fact that most assembly algorithms do require very tightly and well-controlled libraries in order to get reproducible performance. So we really built a solution based on our tunable PlexWell platform, which was really optimized to solve that problem. And as Joanne has mentioned, has been used now for many thousands of plasmids in the Addgene repository. As we, as many other researchers and companies, awoke to the realization a few months ago, certainly the challenge of COVID-19 now was certainly apparent to us, and the urgency it became clear in February and March. And we started to look at ways that we could also help in that area. And certainly looking at genomic tools and NGS multiplexing being at the core of many of the challenges that researchers are facing. And we started to look at opportunities really to address some of the challenges for sequencing that were emerging. Sequencing of SARS-CoV-2 really has a lot of clear impact, for example, in understanding and responding to different types of epidemiological questions. Certainly the tracking of patterns of mutations, identifying modes of spread, and looking at the overall trajectory of the virus as it spreads around the world. It's really important to realize that sequencing is also has a distinct utility that's really different from the diagnostic testing, which is of course critically important. But sequencing provides really an important layer of information in terms of the actual mutations of virus and being able to track those mutations and connect patterns of spread around the world. And we've certainly seen, I'm sure as many of you have in the media, numerous reports where we can make certain assessments and conclusions about how the virus continues to spread. So we, in thinking about that problem, we certainly were aware of the need for multiplex sequencing. And so, we really felt that our PlexWell platform could be a really ideal platform for us to build a more scalable sequencing based approach. Just to say a little bit about PlexWell and what it is, PlexWell is a multiplex library prep platform that we created that really allows you to take large numbers of samples and process them in parallel to make a single library off of multiple samples at the same time. One of the really key attributes of this technology is that it allows you to take input samples that have a range of different concentrations and input amounts, and pull them together after a simple tagging step and produce a normalized library out at the end. Really, this is an effective way of processing, many types of samples where you are interested in multiplexing. And of course, it's really key to getting scalable sequencing. So the workflow is really designed to allow you to do, for example, 96 well plate assays in a matter of a couple of hours, but really also scale up to, in many cases, hundreds or thousands of samples. And this is an example, of our workflow for a 96 well plate, where, as you can see, we take 96 samples and can pull them into a single tube and process that certainly very quickly and with potentially multiple plates at the same time. What this really allows is for what we call scalable multiplexing. The idea behind this is that, again, you can replace what would normally be a library prep process where you're taking individual samples and making individual libraries, and replace that with a process where you can take multiple samples and make a single library off of them at the same time. One of the real features of this, again, is the normalization component, which really saves a lot of time compared to conventional library prep workflows. As you can imagine, there's a range of different applications where this approach is very useful, and we've certainly seen successful application of this in areas such as low-pass whole genome sequencing, single-cell RNA-seq, and as Joanne mentioned, amplicon and plasmid sequencing. Of course, sequencing of viral specimens is certainly well connected to these types of applications, and we approach that from the point of view of having a multiplex PCR foundation, as you'll see in the next few slides. But the key takeaway here is that PlexWell is really a streamlined way of making multiplex libraries very simply and efficiently with improved overall time, cost, and performance. So in thinking about this problem, we were aware of efforts by researchers to create sequencing tools for the SARS-CoV-2 virus. And most notably, a group known as the Arctic Network had created a protocol for doing multiplexed RT and PCR that essentially allowed researchers to take samples and process them through the RT and PCR steps to the beginning point of library prep. And we felt that was a really ideal starting place for potentially applying PlexWell. First of all, because the Arctic protocol, being somewhat well established and used by researchers around the world, was familiar and certainly has been optimized and is something that people are now widely using. So we really felt that this was an excellent place to start, and of course the potential here is again to be able to take that process and plug it into a library prep workflow, which would allow for sequencing of hundreds or thousands of samples at the same time. To say a little bit about the Arctic Network and what it is, it's a consortium of laboratories that have been created. The history goes back to certainly early work by Josh Quick and Nick Lohman on the Ebola virus and Zika virus a few years ago. They have taken that same approach and built it into a sort of a widely used platform for doing multiplexed RT and PCR. Certainly in the last few months, as labs have awoken to the challenge of how to collect information from coronavirus specimens, this has been one of the more commonly used platforms. And so, for example, there have been several publications, a couple of which I note here, that have used this in different library prep workflows. to sequence samples from different types of collections. The typical ARTIC protocol really is again taking the multiplex PCR and combining it with a downstream library prep with different workflows. The most commonly used workflows, again, that had been reported before were generally either ligation-based workflows where one takes the PCR products and simply puts them through a ligation-based library prep. Of course, this comes along with all of the requisite normalization steps. Alternately, certainly in a similar vein, there is a Nextera XT, sometimes miniaturized protocol as well. Again, both of these are really designed to allow individual ARTIC samples to be processed and turned into libraries that can be pooled and sequenced. You know, our goal here was, again, to take and evaluate the potential of taking the ARTIC protocol and using it as part of PlexWell in order to really enhance and really accelerate the speed with which multiple samples could be multiplexed in a single assay in, for example, a few hours. So in looking at this problem, one of the first things we really had to assess was the working range that we would be seeking to achieve. And just to go through a little bit of the background on this, certainly in looking at the data coming out of RT-PCR, the qPCR sort of diagnostic assays, typically, it's been reported that the average viral load in positive samples is in the sort of average in mid-20s, 24 or so CT range. What this really means is, again, our goal was to allow labs that were implementing testing pipelines, we wanted to provide them with a protocol that would allow typical positive samples to be plugged into a sequencing workflow downstream to essentially act as a sequencing method that could be deployed on positive samples. And so in looking at that, we really assessed the working range that was necessary. And for example, in looking at that, we established that really the working range is generally between about 1,000 up to, in some cases, many thousands of copies of virus per sample. And so really our goal then became, okay, can we establish the sequencing performance for a working range that goes down to 1,000 copies of viral RNA into the assay? And that became sort of the baseline goal for our approach. So this is an overview of the protocol that we have then put together based on looking, again, taking the ARTIC protocol and making some slight modifications and combining it with PlexWell. I'll note, as you see in the lower right here, that we have made this protocol available on our website. You're welcome to go and review that and read it in more detail. But the key features here really are, again, showing, and again, you'll see data in the next few slides on examples where we can take sample human nucleic acid samples with spiked-in controls variously from either the twist or ATCC references, input that as the beginning for the RT and multiplex PCR part of the ARTIC protocol, and then without actually needing any purification, go directly into PlexWell and make the multiplex libraries that can be loaded directly onto sequencers. So the next few slides, I'll be presenting data that sort of go through the results that you achieve using this protocol. So one of the first things we really were interested in looking at here was, can we achieve high coverage libraries that that are sufficiently well covered even with low viral copy number. What you can see here is again a range of viral copy numbers going into the RT and multiplex PCR, and then put into PlexWell, and that the amount of data you get back certainly tracks with the amount of virus that goes in, but certainly down to a reasonable limit of 800 copies or so. We have sufficiently high high coverage to say that we're likely going to be able to confidently call variants in those viral genomes. And that's really borne out in this next plot that I'm showing, where you see the percentage of the actual SARS-CoV-2 genome that's covered at different coverage levels, 1x, 10x, and 30x, depending on the number of viral copies that went into the RT and multiplex PCR. So again, this gives again another view of the data, which shows that above a number of viral copies, essentially in the mid-hundreds, certainly to 1,000. Above 1,000, we have uniformly high coverage of samples going into the assay. And again, as that sort of, again, needs to play out, of course, is how does that look at, how does that translate into variant calls? And what you can see here is the sensitivity, essentially our ability to detect the known variants in those samples. For example, there are 3 variants that are present in the ATCC reference versus the original Wuhan strain. And our ability to detect those and accurately call them both positively and negatively also tracks very well with coverage. So certainly up to, again, a threshold of 800 copies, we have 100% sensitivity and specificity for those variants. Again, the uniformity of performance, again, in that working range, again, our goal here was to be able to really see that we were getting predictable and uniform performance across a wide range of viral copy number. And as you can see here, ranging from as many as 100,000 copies down to 800, you see, again, uniformly high coverage across the full SARS-CoV-2 genome and mapping rates that are well above 80%. So this certainly gives you an illustration of the performance that can be achieved here, combining the Arctic protocol with PlexWell. Of course, the other advantage that really PlexWell brings to this problem is the ability to scale it up. And so what we then looked at is really a sort of larger scale study where we were going to take a large number of samples and multiplex this to make sure that we got uniform performance in a much larger multiplex context. So in this case, what we did is we took a single input plate with a range, again, of different viral input amounts, again, across our full working range, and made 4 replicate 96 plex libraries, which you see illustrated in terms of their raw read counts here. And then, again, looking here at the ability to cover each of those genomes at some minimal level that would be required to call variants. And the performance there, again, was quite good, especially if you factor in, again, several orders of magnitude difference between the low and high amount of viral RNA going into the assay. The actual amount of data coming out at the end was actually quite good and uniform. So this really is, again, a good example of how one could scale this assay up. And as I'll mention in the next couple of slides, we do have PlexWell assay configurations that can support up to 1,152 samples in the same run. So just to say a few words in summary here about the Arctic and PlexWell protocol, and again, our work here. Certainly the key findings from this work is that we can achieve robust performance, looking at the protocol being able to make high quality libraries in the background of human nucleic acid, which would be needed for full genome sequencing of the coronavirus genome. Certainly an improved and simplified workflow compared to other methods without the need, for example, of normalization or purification of the amplicons going into library prep. We feel like this is a key feature that will allow this protocol to be deployed more quickly and easily in labs. And again, establishing a working range which is matched to the likely number of viral RNA genome equivalents that would be going into this assay in the context where it's run on samples that were tested positive via the sort of standard RT-PCR assays. So again, that working range is certainly supports that you're going to be able to get high quality genome coverage down to the sort of low end of what RNA copy numbers of positive samples would generally be. And of course, one of the primary benefits of PlexWell that certainly applies here as well is the ability to take, again, a simple workflow and scale it up. So again, regardless of what type of sequencer you might have in the lab, whether it be a MiSeq or a NextSeq or even a larger sequencer, we have different kit configurations that can support, up to 96 samples per run, or multiples of 96 or 384 samples per run. So again, it gives that flexibility in terms of the number of samples that can be loaded onto the sequencer and run in your lab. So in conclusion, really, I think the advantages of this method are robust performance, simple workflow, and the high multiplexing capabilities. I should also note this is because of the fact that we're saving for example, in certain parts of the workflow that would otherwise be needed, such as normalization. This is a very cost-effective solution. Being able to make a single library from many samples takes a lot of steps out of what would otherwise be usually a more difficult library prep workflow. And so this really is going to translate into significant cost savings overall versus many other methods. It's also automation friendly. Again, we certainly don't require automation to use the PlexWell process, but it certainly is friendly to automation. And in fact, we're currently collaborating with Opentrons on an open source protocol here to implement this and distribute it to researchers around the world. And it would certainly be easily amenable to automation on other platforms. And we have experience seeing PlexWell automated on Biomech and TCAM platforms, for example. In terms of future directions and other areas we're working, I will just say a few notes about this. Yeah, certainly in terms of the multiplexed RT and PCR process, I think there's certainly some improvements that we're very interested in exploring there and are working on. For example, simplifying the workflow, improving the performance and limit of detection of the assay, and really saving some of the time that's associated with that part of the workflow. And of course, in the scalability side of things and being able to really have this assay perform, at larger scale and even better, we are looking at additional barcoding index reagents and looking even at the ability to support unique dual indexing as part of this protocol in order to really get the best performance for a range of different assay needs. So with that, I'll conclude here. Again, I appreciate everybody joining us here, and I'm certainly happy to take questions. I'll just put notes here that if you had any questions about the protocol or certainly the workflow that we presented here, I certainly invite you to visit our application support page at SeqWell.com. And of course, if you want to learn more about the tools that Addgene has created, addgene.org will have all the information that you'd be looking for as well. So thank you very much. So if there are any questions, I will certainly be happy to take those at this time. So one of the questions that came in on Q&A here, and I'll read through a couple of these. There's a question here regarding the spike in control from ATCC or twist. So what we have done here, the question is it spiked into the sample? Is it a limit of a blank control? So we took the ATCC and twist samples and tested both of them as spike-ins to a total nucleic acid purified sample at different, essentially different titrations to assess the performance of the assay for different amounts of virus that would be in a typically encountered scenario there. Next question I see here is how this is different from COVID-seq that was just announced by Illumina a couple of days ago. So, as you may be aware, the COVID-seq assay is a diagnostic assay that is meant to, in a way, sort of replace or allow a more scalable form of the RT, what would otherwise be done with RT-PCR. It doesn't actually sequence the full genome, so it's really designed as a quantitative assay to really assess the specific amount of virus that's in a sample, but it doesn't actually give you the full sequence information of that virus. Here's a question for, I think, let's see, we have a question here. How can you increase the sensitivity and sequencing and pooling of the workflow? Have you seen false negatives? So because this is really not designed as a diagnostic assay, our focus has really been on achieving good sequencing performance in a range of RNA amounts that would be coming in, in samples that would have typically been tested positive, kind of upstream. It's a good question, though. I mean, again, I think you know, as you see again with the use of sequencing as a sort of do everything assay, if you can improve the sensitivity of the assay in order to you know, be able to detect lower and lower amounts of virus, then certainly including quantitative controls, I think certainly would be a good way of being able to, you know, make this a more quantitative assay. But our focus right now has been really on getting uniformity of sequencing coverage, so that, you know, one can use this to get, you know, reproducible, high quality genomes from samples that you have sort of independently tested to actually contain virus. Another question here, can we elaborate on customers who are currently using PlexWell for COVID-related sequencing? And does it work for custom amplicon panels or fewer than 96 samples? So certainly I can say a few things about that without you know, sharing information from some of our that would be, you know, kind of more confidential. But I think, you know, we do have some of our users who are using this for different types of amplicon sequencing workflows in the COVID application space. Again, it ranges from, you know, sort of different sizes of amplicons and different numbers of amplicons in that assay. In terms of custom amplicon pools and panels, absolutely. In fact, I think, you know, I think this is a real great area for exploration here is, you know, coming up with different panels and different content that can be used as part of this protocol. And then as to whether it can be used on smaller numbers of samples, absolutely. We actually have a kit called PlexWell Plus, which is really designed to handle, you know, lower multiplexing batches from between 8 and 96 samples. I have a question here from Fritz Roth. Hello, Fritz. Fritz says, thanks, Joanne and Joe, for your efforts on SARS-CoV-2 plasmid sharing and genome sequencing. Question is, it seems like one of the major advantages of PlexWell beyond indices is skipping normalization. Can we say more about how that works? So yeah, I'm happy to do that. If we go back to our workflow, I can actually share the slides here. It will be easier to talk through this live. The key to this process really is this iterative barcoding procedure, taking sample barcodes and tagging samples in the first step with a equal amount of sort of different barcoded reagents in a 96-well plate. What that does is it effectively tags those samples and creates the same number of tagging instances that once those samples are pooled, allows you to make a library in which, as long as you've got the same number of tags in each of those samples, you're actually tolerant to different amounts of DNA. So it's that sort of limitedness of the sample barcoding step combined with pooling and making a library off of that pool, which effectively gives you that normalization performance. Question here from Randy Kerstetter. Hello, Randy. He asked, would we comment on the analysis that is done at SeqWell? So I'm going to infer from this is this maybe I'm going to interpret from this he probably is referring more to the plasmid analysis pipeline that Joanne mentioned earlier, and not the sort of standard bioinformatics pipelines that we've been running on the COVID data that we've been generating. We've created a very tuned algorithm that's really designed to take advantage of some of the features of PlexWell libraries in terms of the expected size and diversity of the libraries that we have optimized the protocol with Addgene to use on plasmid libraries. And I think that tuning and combined with, again, you know, the tight control of the libraries has been the key there. Without getting too far into the weeds on the actual steps of the analysis, it uses sort of standard, you know, De Bruijn graph type assemblers, using, you know, different pre-processing steps to make sure that we don't put too many kind of noise-containing reads into the assembly. And then we do a remapping process after assemblies are made in order to go back and make sure that we can get confident read calls throughout a fully assembled plasmid. Final question I see here from one of our attendees is just the cost per sample. So first of all, I will say, as it relates to the work in COVID. We are actually, again, if you go to our website, there is a promotion that we're doing here to really enable researchers who are trying to solve different types of problems in the sequencing of COVID samples or SARS-CoV-2 genomics. I think that's really the first place I would direct you. In general, our kits are priced very competitively and really designed to allow you to hopefully process more samples, saving essentially saving more per library. And certainly, I would encourage you to reach out and follow up with one of the members of our great commercial team to discuss that more. One of the other questions here is again, mentions template switching can be an issue for applications with multiplex libraries, where barcodes can be connected to other barcodes in a library. Do we have diagnostics in our analysis pipeline that can detect and remove template switching events that can otherwise cause mismapping between libraries? Yeah, so this is a great question, and in fact, I think there's a couple of detailed issues there that that certainly would require in-depth discussion. In general, what we find for most mapping applications, again, the rate of template switching that we observe in these libraries, which are again large complex libraries that have many barcodes present, is far below the amount that would generally be introduced in any kind of standard mapping or variant calling pipeline. And by controlling the amount of that switching below a certain level, which was part of the optimization process of how we have created these kits, we certainly are confident that that is not a problem that is going to cause false variants, for example, to be detected. And again, it can also be approached as a sort of threshold setting problem where you simply have to know the amount of that template switching and essentially remove or ignore any reads that might be observed at or below the expected level, which in our typical, again, typical assays that we've looked at is below 1% in any library. So again, that's a succinct answer to a somewhat complicated question that I'm sure, if you reach out, we would be happy to talk about more. So I think there's, I don't see any other questions here. If we, maybe we could conclude here. I think we're running close to our allotted time. But certainly, I think we really appreciate everybody's attendance here today, and it was great to have the ability to talk about the work we're doing, and certainly hear about the work Addgene is doing to support the distribution of tools for a variety of applications, including COVID-19 research.